Hi folks, and welcome to another edition of Zog Science. Today we're going to be taking a look at transcription and translation and the role they play in protein synthesis. So transcription is the first process in protein synthesis, and it is DNA-directed synthesis of RNA. RNA synthesis is catalyzed by a protein called RNA polymerase, which is an enzyme. And it is going to peel apart the DNA strands and join together the RNA nucleotides. It is only able to assemble nucleotides in the 5' to 3' direction, just like DNA is only able to be synthesized in the 5' to 3' direction. However, unlike DNA polymerases, it can start making a chain without a primer, which makes sense if you think about the fact that in order for DNA polymerase to work, we had to first add the RNA polymerase, or the RNA primer, which was added by a primase, which didn't have anything to add that on to. So your RNA can be made without the primer. Here's an animation that's going to kind of introduce us to the transcription process.
The central dogma of molecular biology, DNA makes RNA, makes protein. Here the process begins. Transcription factors assemble at a specific promoter region along the DNA. The length of DNA following the promoter is a gene, and it contains the recipe for a protein. A mediator protein complex arrives carrying the enzyme RNA polymerase. It maneuvers the RNA polymerase into place, inserting it with the help of other factors between the strands of the DNA double helix. The assembled collection of all these factors is referred to as the transcription initiation complex, and now it is ready to be activated. The initiation complex requires contact with activator proteins, which bind to specific sequences of DNA known as enhancer regions. These regions may be thousands of base pairs distant from the start of the gene. Contact between the activator proteins and the initiation complex releases the copying mechanism. The RNA polymerase unzips a small portion of the DNA helix, exposing the bases on each strand. Only one of the strands is copied. It acts as a template for the synthesis of an RNA molecule, which is assembled one subunit at a time by matching the DNA letter code on the template strand. The subunits can be seen here entering the enzyme through its intake hole, and they are joined together to form the long messenger RNA chain snaking out of the top.
All right, so here are the three steps that are involved in the process of transcription. You have initiation, elongation, and termination. In initiation, we are going to be beginning the process of producing the RNA transcript using the RNA polymerase enzyme. The DNA is going to be unwound, right, in order for that to happen. Then the DNA is rewound, and the RNA polymerase will move along the gene, and it will continue to produce the RNA transcript. The direction of transcription is considered downstream, and the opposite direction is considered upstream. And eventually, once the RNA polymerase reaches the end of the sequence, it is going to pop off, and the completed RNA transcript will be released. Again, the sequence where the RNA polymerase attaches is called the promoter region. In bacteria, the end of this transcription process, the end of the transcription unit is called the terminator. And the stretch of the DNA that is transcribed, that is called the transcription unit. The promoter region is used in both eukaryotic and prokaryotic cells. Again, the three stages of transcription are initiation, elongation, and termination. And the promoter signals the transcriptional start point, and that is where initiation will begin. Transcription factors and mediator proteins are going to help the binding of RNA polymerase and will initiate the process of transcription. In our next video, I'll talk about a little bit more about that process and how it is regulated in eukaryotic and prokaryotic cells. The full process or the full assembly of all of the transcription factors and other proteins is called the transcription initiation complex. And in eukaryotic cells, this is forming around the TATA box, which is inside the promoter region. So here is the TATA box, which is where some transcription factors are going to bind, and then the RNA polymerase is going to be able to bind at this location using the transcription factors to kind of help with that process, and then transcription will begin. As RNA polymerase moves along, it is untwisting the DNA helix and it is adding in nucleotides. And a gene can be transcribed simultaneously by several RNA polymerases. So once one gets farther enough along, it can begin to be transcribed by a new RNA polymerase. Here's kind of an inner picture showing you the direction of transcription. So you'll notice that the DNA strand is being read from the 3' end to the 5' end. The RNA is being made from the 5' end to the 3' end. And the reason, again, is that you must add nucleotides onto the 3' end of a DNA or RNA molecule because it has the free hydroxide group for the dehydration synthesis reaction to take place. So that's the reason why it's moving in the 5' to 3' direction of transcription. The mechanisms of termination are different in bacteria and eukaryotes. In bacteria, the polymerase stops transcription at the end of the terminator. So when it gets to the terminator region, the mRNA stops translating, and that's it. In eukaryotes, the DNA polymerase transcribes a polyadenylation signal sequence, which is at the end, and then the RNA transcript is released 10 to 35 nucleotides past this sequence. Now, in eukaryotic cells, RNA is modified after transcription. This is a very important process. You want to make sure that you understand what's going on. Prokaryotes do not modify their RNA, but eukaryotes do. At each end of the pre-mRNA molecule, which is what it is called when it is first transcribed, the 5' end receives a modified G nucleotide 5' cap, and the 3' end gets a poly A tail. The 5' cap helps the ribosome to be able to attach at the 5' end. It also protects the mRNA and helps it to be transported out of the cytoplasm. The poly A tail helps to protect the mRNA from hydrolytic enzymes, which we learned about are the exonucleases. So here is what a mRNA molecule will look like after it has been modified. It's got a 5' cap and it's got a poly A tail. Here is that polyadenylation signal that you can see here, and there are these regions before and after the start and stop codons that are called the UTRs, and those are just kind of areas that provide a little bit of buffer on either end in case something were to go wrong. Now, most eukaryotic mRNAs have long non-coding stretches of nucleotides that lie between the coding regions. You can see those here in the lighter shades. These non-coding regions are called introns, or intervening sequences. The other regions are called exons, and they are what are actually used to create the amino acid sequences. So RNA splicing is a process where we remove the introns and join the exons together in order to form the mRNA molecule that is actually going to be sent to the ribosome for translation. So here is that process shown a little bit more up close. The introns are going to be removed. The exons are going to be kept, and that is going to leave you with your actual coding segment. Notice that the UTRs are kept, as well as the 5' caps and the poly A tails. Because of the fact that you have multiple exons, you can splice different exons and keep some of them and get rid of others, and this is called alternate RNA splicing. And alternate RNA splicing allows for multiple polypeptides to be created by a single gene. RNA splicing is carried out by things called spliceosomes, which are made up of proteins and small RNAs. And these RNA molecules are sometimes called ribozymes because they function as enzymes. And we will see that the ribosome is a type of ribozyme. So we've got our modified mRNA, or if it's a prokaryotic cell, our just regular mRNA, and now it's going to be actually translated into a protein, and that process is going to take place at the ribosome. So a cell translates the mRNA message into protein with the help of a transfer RNA. A transfer RNA, or tRNA, is transcribed just like other RNA molecules, but rather than carrying a message from the nucleus to the ribosomes in the cytoplasm, it is going to just be folded and changed into a tRNA molecule. tRNAs carry amino acids or transfer amino acids to the growing polypeptide chain in the ribosome. So we're watching another little video to kind of be able to see what's going on with the ribosome and the translation process.
The job of this mRNA is to carry the gene's message from the DNA out of the nucleus to a ribosome for production of the particular protein that this gene codes for. There can be several million ribosomes in a typical eukaryotic cell. These complex catalytic machines use the mRNA copy of the genetic information to assemble amino acid building blocks into the three-dimensional proteins that are essential for life. Let's see how it works. The ribosome is composed of one large and one small subunit that assemble around the messenger RNA, which then passes through the ribosome like a computer tape. The amino acid building blocks, that's the small glowing red molecules, are carried into the ribosome attached to specific transfer RNAs. That's the larger green molecules, also referred to as tRNA. The small subunit of the ribosome positions the mRNA so that it can be read in groups of 3 letters known as a codon. Each codon on the mRNA matches a corresponding anticodon on the base of a transfer RNA molecule. The larger subunit of the ribosome removes each amino acid and joins it onto the growing protein chain. As the mRNA is ratcheted through the ribosome, the mRNA sequence is translated into an amino acid sequence. There are 3 locations inside the ribosome, designated the A site, the P site, and the E site. The addition of each amino acid is a 3-step cycle. First, the tRNA enters the ribosome at the A site and is tested for a codon-anticodon match with the mRNA. Next, provided there is a correct match, the tRNA is shifted to the P site, and the amino acid it carries is added to the end of the amino acid chain. The mRNA is also ratcheted on 3 nucleotides, or 1 codon. Thirdly, the spent tRNA is moved to the E site and then ejected from the ribosome to be recycled. As the protein synthesis proceeds, the finished chain emerges from the ribosome. It folds up into a precise shape determined by the exact order of amino acids. Thus, the central dogma explains how the 4-letter DNA code is, quite literally, turned into flesh and blood.
All right, so let's look at that process in a little bit more detail. So we have our ribosome and we have our tRNA. tRNAs have the amino acids attached to them. The codons are on the mRNA that is pulled through the ribosome, and we're looking for anticodons on the tRNA that will be complementary to the codons on the mRNA. When they match up, they bring their amino acids with them, and they are dropped off, and they form the polypeptide chain. So each tRNA can translate a particular mRNA codon into a given amino acid, which is why it's called translation. The tRNA contains an amino acid at one end, and on the other end it has an anticodon that is complementary with the codon in the mRNA. So this is what the tRNA looks like. Here is the anticodon. There are hydrogen bonds between the bases that holds it together and gives it its kind of T shape, which is why it's another reason why it's called tRNA. And it has the anticodon, again, that is going to match up. Ribosomes are what facilitate the specific coupling of the tRNA anticodons with the mRNA codons. And remember that ribosomes are made up of rRNAs, and there are 2 subunits, the large and small subunits. In bacterial and eukaryotic ribosomes, the large and small subunits form together to actually form the ribosome, but they only do that when they are attached to an mRNA molecule. So here is what the ribosome looks like. You can see the A, P, and E sites, right? So the A site, that is the where the tRNA comes in. The P site is where the polypeptide chain is made, the amino acids are joined together, and then the E site is where the tRNAs are exited. And that's what I just talked about. So the 3 stages of translation are also initiation, elongation, and termination. So some similarities there. All 3 stages require protein factors that aid in this process, and the energy comes from the hydrolysis of GTP rather than ATP. The initiation stage of translation brings together the mRNA, a tRNA with the first amino acid, and the 2 ribosomal subunits. We know that the starting amino acid has got to be methionine because that is going to match up with the start codon, which is AUG. So the mRNA is found until it reaches the AUG, which is going to match up with the methionine. And the A sites, then the large subunit is going to come in and add in the A site. The P site will go directly where that tRNA is, and then the E site is left there. The start codon is important because it establishes the reading frame for the mRNA. That is the sequence of the 3 nucleotides for the reading. And the addition of the large ribosomal subunit is going to complete the initiation complex, and translation will begin from that time. During elongation, amino acids are added on one by one using the anticodon-codon pairing, and the translation is going to be reading the mRNA in a 5' to 3' direction. So during elongation, we have our one amino acid that has been added, and it is beginning to make the polypeptide chain. The tRNA is bound to the P site, and a new one is coming in that is going to bind the anticodon with the codon. We then have the next one, which is the actual bonding is going to occur in the P and A sites. So the polypeptide is going to be added onto, the new amino acid will be added onto. Then we have translocation, so the polypeptides are going to move. The mRNA is pulled to the next site, so the tRNA is moved to the next site. The used tRNA is ejected, and the next tRNA is left behind, and this process will repeat. Termination occurs when we finally get to a stop codon, and the stop codon is going to bind to a protein called a release factor. And the release factor causes the addition of a water molecule instead of an amino acid, and this causes the release of the polypeptide, and the translation and the ribosome is going to come apart. So we have our stop codon, which are either a UAG, UAA, or UGA. That's going to have a release factor. The release factor is going to release the polypeptide with an extra water on it, and then these ribosomal subunits are going to pop apart. Often translation is not sufficient to make a functional protein, and polypeptide chains are often modified or translated after translation, or they're targeted to specific sites in the cell. We're not going to talk about that specifically at this point, but there are usually some steps that happen after that. During synthesis, a polypeptide chain is going to begin to coil into its 3-dimensional shape because that happens automatically. That's not going to be something that requires specific energy to take place, but it does happen automatically. But there are other places where the other post-translational modifications can occur. Now, in bacteria and eukaryotes, multiple ribosomes can translate a single mRNA at one time. Once a ribosome is far enough past the start codon, another ribosome can attach, and strings of ribosomes are sometimes called polyribosomes. You can kind of see that process here. You've got a piece of mRNA, and then you've got all these ribosomes that are being attached. Here is an example of an mRNA with some ribosomes attached in a bacterial cell. Now, bacteria and eukaryotes can also transcribe multiple mRNAs from the same gene. So that means that transcription and translation can take place simultaneously in bacteria, whereas in eukaryotes, the nuclear envelope separates those two processes. So here is in a bacteria, here is the DNA. Here is the RNA polymerase that is creating the mRNA strand. Here are a bunch of polyribosomes that are all making proteins at the exact same time. So you can create lots and lots of copies all at once. In a eukaryotic cell, these processes are separated because of the nucleus. So recall that we have transcription, which is the formation of the RNA strand. The mRNA is then going to be processed. The processed mRNA is then going to be sent out to the ribosomes, and then the process of translation can begin. All right, that's all I've got for you this time. Thanks so much for watching, and I'll see you next time.